8 weeks. That is the extraordinary amount of time it took Oxford scientists to develop a brand new Ebola vaccine and move it all the way into human trials. As of this week, the United Kingdom is at the center of one of the most remarkable medical stories of 2026, with the first ever human trial of a Bundibugyo Ebola vaccine now underway. Ebola kills between 25 and 90% of the people it infects, depending on the strain and the quality of medical care available. It can cause uncontrolled internal and external bleeding, organ failure, and death within days of symptom onset. The fact that a vaccine targeting a specific strain of this virus was designed, manufactured, and cleared for human testing in just 8 weeks is genuinely unprecedented in the history of vaccine development. Stay with us as we break down exactly what is happening, why it matters, and what comes next. To understand why this trial is so significant, we need to look at how this emergency unfolded. The United Kingdom declared an Ebola emergency, and within 8 weeks of that declaration, Oxford University had produced a working vaccine candidate ready for human testing. The virus at the center of this outbreak is the Bundibugyo strain, a particularly dangerous variant that previously caused outbreaks in Central and East Africa, including in the Democratic Republic of Congo in 2012. The broader emergency prompted urgent responses from health authorities across multiple countries. Encouragingly, as of the 16th of July, 2026, Uganda began its 42-day countdown to being formally declared free of the virus, suggesting that containment efforts on the ground have shown some early success. But the race is far from over. With human trials now underway in Britain, the next phase of this fight has entered a critical new chapter, and the world is watching closely. How do you build a vaccine in 8 weeks? Normally, vaccine development takes years, sometimes decades. Regulatory approval alone can stretch out across years of clinical data and safety reviews. So how did Oxford's team move at such remarkable speed? The answer lies in a combination of pre-existing platform technologies, emergency regulatory frameworks, and the hard lessons learned from previous outbreaks. Oxford researchers had already been working on viral vector vaccine platforms, the same approach used to develop the COVID-19 vaccine that Oxford produced alongside AstraZeneca. This means the underlying technology was not built from scratch. Instead, scientists adapted a proven delivery platform to carry the specific antigens of the Bundibugyo Ebola virus. Emergency regulatory pathways, refined significantly during the COVID-19 pandemic, allowed expedited review without compromising safety standards. The result was a vaccine candidate ready for its first human trial in a timeframe that would have seemed impossible just a decade ago. The Bundibugyo strain of Ebola is not the best-known variant of the virus. Most people associate Ebola with the Zaire strain, which caused the catastrophic 2014 to 2016 outbreak in West Africa that killed more than 11,000 people. But Bundibugyo is no less serious. It was first identified in Uganda in 2007 and has since caused significant outbreaks in Central Africa. Like all Ebola strains, Bundibugyo infection begins with fever, severe headache, muscle pain, and sore throat, before progressing to vomiting, diarrhea, and rash. In the most serious cases, patients experience internal and external hemorrhage as the virus attacks the body's clotting mechanisms. There is currently no fully licensed vaccine specifically for the Bundibugyo strain, which is precisely why this Oxford trial is so important. Until this week, the world had no validated medical countermeasure targeting this variant directly, leaving entire populations exposed with no specific protection. The trial itself is taking place in the United Kingdom and represents the very first time any human being has received this particular vaccine. Early-phase clinical trials like this one are primarily designed to assess safety and immune response, rather than to prove full efficacy. Volunteers who receive the vaccine will be monitored closely over the coming weeks and months for any adverse reactions. Researchers will also measure how strongly their immune systems respond to the Bundibugyo antigen. If the results are promising, the trial can progress to larger Phase 2 and Phase 3 studies involving thousands of participants, ideally conducted closer to active outbreak zones where real-world efficacy can be properly assessed. Researchers at Oxford described the current situation as brutal, painful, and very distressing. That language is striking coming from scientists, and it underlines the urgency driving every decision in this program. This is a genuine medical emergency being answered with genuine scientific speed. The impact of this Ebola emergency extends well beyond laboratories and hospital corridors. For communities affected by active outbreaks, daily life is shaped by fear, isolation, and grief. Ebola spreads through direct contact with the bodily fluids of infected individuals, which means that the act of caring for a sick family member carries an enormous personal risk. Healthcare workers on the front line are among the most vulnerable, as the 2014 West Africa outbreak tragically demonstrated. In Uganda, where the 42-day countdown to a virus-free declaration began on the 16th of July, 2026, there is cautious optimism. But as health authorities know from painful experience, that countdown can be reset if even a single new case emerges. The vaccine trial underway in Britain is therefore not just a scientific exercise. For the nurses, doctors, and community health workers operating in affected regions, it represents a potential lifeline at a moment of profound vulnerability. Where does this go from here? If the Phase 1 trial data looks strong, Oxford's team could move quickly into expanded trials, potentially in affected regions of Africa within months. Emergency authorization frameworks developed during the COVID-19 pandemic have created a much faster path to deployment than existed before. Regulatory bodies in the UK and internationally can now act with speed without sacrificing rigor. Global health organizations have long called for a stockpile of Ebola vaccines covering all 4 human pathogenic strains. Currently, approved vaccines exist for the Zaire strain, but not for Bundibugyo. If this trial succeeds, it would fill one of the most significant gaps in the world's pandemic preparedness toolkit. The timeline from here to potential deployment is still measured in months to years, not weeks. But today, on the 21st of July, 2026, the clock is ticking in the right direction, and the scientific community has good reason to be cautiously optimistic. 8 weeks to build a vaccine. A deadly virus with a mortality rate that can reach 90%. A global community watching and waiting. The story of the UK's Ebola vaccine trial is about far more than one clinical study. It is a demonstration of what modern science is capable of when urgency, expertise, and political will align at precisely the right moment. Oxford University and the broader UK research community have shown that the world does not have to be defenseless in the face of a sudden viral emergency. The results of this trial will not be known for some time, but the fact that it is happening at all, just 8 weeks after an emergency was declared, is itself a form of hope. If you found this video informative, please give it a thumbs up and subscribe to our channel for more in-depth coverage of the biggest health stories shaping the world in 2026. We will keep you updated as the trial data comes in.